Glycolysis.
Good day, everyone. Today we'll be introducing glycolysis, a vital process in cellular metabolism. Glycolysis is the pathway through which glucose, a simple sugar, is breaking down into pyruvate, producing energy in the form of ATP and NADH. It takes place in the cytoplasm and is essential for both aerobic and anaerobic respiration. Let's dive deeper into how this process works.
In the first step of glycolysis, glucose enters the cell cytosol and it's converted to glucose 6-phosphate by a hexokinase enzyme. Magnesium, a metallic element, weakens the phosphodiester bond of ATP, allowing phosphate to be released and transferred to a glucose in the 6-carbon. This reaction involves a nucleophilic attack of the C6 hydroxyl, promoting a favorable conformation of ATP at the enzyme active site. The phosphorus-oxygen bond is weakened by the magnesium, allowing phosphate to be removed and transferred to glucose.
Step 2 in glycolysis. So, in this step, the enzyme that catalyzes is phosphoglucose isomerase with the help of enzyme glucose 6-phosphate and produces fructose 6-phosphate. So, these steps promotes isomerization or rearrangement reaction, and this step is reversible. Step 3. So to continue, in the third step of glycolysis, this is an irreversible type of reaction and there are 2 ADP used. So this is a phosphorylation reaction with the help of enzyme fructose 6 ATP and produces the product which is fructose 1,6-bisphosphate plus ADP. It is catalyzed by phosphofructokinase and as you can see, Mg or magnesium is always present. And its purpose is to weaken the phosphodiester bond so the phosphate would be easily released. This step is the slowest out of 10 steps, which means enzymes are highly regulated and it is a highly spontaneous reaction.
So the aldolase catalyzes the hydrolysis of fructose 1,6-biphosphate into 2 different molecules, and these 2 different molecules have 3 carbons each, which are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, meaning to say that the fructose 1,6-biphosphate is cleaved, hence the aldol cleavage reaction, as the enzyme aldolase aids in this process. The enzyme triose phosphate isomerase catalyzes the conversion of DHAP to glyceraldehyde 3-phosphate. Now this process occurs as DHAP cannot proceed to the next step by itself. Therefore, it has to be converted into glyceraldehyde 3-phosphate. In the step-by-step process, the enzyme TIM binds to DHAP, in which the TIM's active site provides an efficient active site. This active site stabilizes the transition state. Now, proton transfer is the reaction involved in this process, which is facilitated by the amino acid histidine, which acts as a proton donor or acceptor during conversion. The isomerization proceeds through an enediol intermediate. Now in this state, the carbonyl group of DHAP is converted into a hydroxyl group, while one of the hydroxyl groups on DHAP becomes a carbonyl group. In the conversion to GAP, the enediol intermediate rearranges to form glyceraldehyde 3-phosphate or GAP. Now this step effectively converts the ketone structure of DHAP into an aldehyde structure of GAP.
Step 6 and 7 in glycolysis. In this stage of glycolysis, glyceraldehyde 3-phosphate undergoes 2 reactions. First, glyceraldehyde 3-phosphate undergoes oxidation. The aldehyde group is changed into a carboxylic acid, and NAD plus is reduced to NADH. Next, GAPTH adds an inorganic phosphate from the cytosol to the oxidized molecule, producing 1,3-biphosphoglycerate. This compound features phosphate groups attached to both the first and third carbon atoms. Step 7 of glycolysis. In these steps, the enzyme phosphoglycerate kinase transfers a phosphate group from 1,3-biphosphoglycerate to adenosine diphosphate, which produces adenosine triphosphate and 3-phosphoglycerate. This process shows substrate level of phosphorylation. These steps work together to increase the net energy gain in glycolysis, allowing the cell to efficiently convert glucose into useful energy.
Step 8 is the phosphoglycerate mutase. This step is all about rearranging the phosphate group on the molecule to get it ready for the next reactions. The enzyme that makes this happen is phosphoglycerate mutase. It has a special active site with a histidine residue that helps transfer the phosphate group. So first, the enzyme temporarily adds a phosphate to the second carbon group of the molecule, creating an intermediate called 2,3-biphosphoglycerate. Then, the enzyme removes the phosphate from the third carbon. The result is 2-phosphoglycerate, a molecule that's now in the correct position for the next step, which is the dehydration reaction in step 9. So the reaction is called a mutase reaction, which means a functional group is shifted from one position to another.
Step 9 in glycolysis. Enolase is an enzyme that catalyzes the conversion of 2-phosphoglycerate or 2-PG to phosphoenolpyruvate or PEP in the 9th step of glycolysis, but it passes first to the enolate intermediate. This reaction is the removal of water. One hydrogen is removed and one hydroxyl is removed. These two will combine to form water. This is the dehydration reaction between phosphoglycerate and phosphoenolpyruvate that helps set up the final energy yielding reaction in glycolysis. In the final step, pyruvate kinase is formed. This step involves another instance of substrate level phosphorylation, where phosphoenolpyruvate transfers a high-energy phosphate group to an ADP molecule, producing ATP and pyruvate.
Pathways of pyruvate.
In step 1, a carboxyl group is removed from pyruvate, releasing a molecule of carbon dioxide into the surrounding medium. The result of step in a 2-carbon hydroxyethyl group bound to the enzyme or the pyruvate dehydrogenase. This is the first of the 6 carbons from the original glucose molecule to be removed. This step proceeds twice. Remember, there are 2 pyruvate molecules produced at the end of glycolysis. For every molecule of glucose metabolized, thus, 2 of the 6 carbons will have been removed at the end of both steps. In step 2, NAD plus is reduced to NADH. The hydroxyethyl group is oxidized to an acetyl group, and the electrons are picked up by NAD+, forming NADH. The high-energy electrons from NADH will be used later to generate ATP.
Pyruvate step 3. In step 3, coenzyme A, an organic molecule generated from vitamin B5, is joined to the oxidized 2-carbon molecule and acetyl group, the one that is highlighted in green, to form acetyl-CoA. The formation of acetyl-CoA, also known as the carrier molecule, is to transport the acetyl group to the citric acid cycle. An important molecule in metabolism, acetyl-CoA serves as a link between the breakdown of nutrients and production of energy. It is essential for anabolic, building chemicals, and catabolic, breaking down molecules processes. The cell could not effectively generate energy or manufacture vital macromolecules without acetyl-CoA. To conclude, a crucial stage that links glycolysis and the citric acid cycle is the oxidation of pyruvate. Acetyl-CoA, CO2, and NADH are produced as a result, and these are necessary for ATP synthesis and cellular energy metabolism. And that's the end of our topic. Thank you.
The Krebs cycle.
Greetings everyone, my group members and I will be explaining the Krebs cycle steps. So what is Krebs cycle? So the Krebs cycle is a fundamental metabolic process that takes place in the mitochondria of cells. It is also referred to as a citric acid cycle or the tricarboxylic acid or TCA cycle. It is essential to cellular respiration, which is how living things produce energy. The Krebs cycle is essential to the metabolism of almost all aerobic organisms because it not only drives the synthesis of energy but also supplies essential intermediates for other metabolic pathways. So step 1 is a glycan condensation. The glycan condensation occurs in the beginning phases of the Krebs cycle. In this step, involves the combining of 2 molecules, acetyl-CoA, which has 2 carbon atoms, and oxaloacetate, which has 4 carbon atoms. Together, they produce citrate, a new 6-carbon chemical. Citrate synthase is an enzyme that helps with this process. Imagine that the cycle begins when a building block, which is the acetyl-CoA, binds to a base oxaloacetate to form a larger structure, which is the citrate, together with oxaloacetate. The 2 carbon atoms donated by the acetyl-CoA form the 6-carbon citrate. The Krebs cycle, which is essential for producing energy for the cell, embarks on the stage, which produces energy in the form of ATP, FADH2, and NADH. Is initiated by this condensation reaction. The cycle would not continue and the cell would not be able to generate the energy required for survival and appropriate operation if citrate had not been formed in this step.
Next, cis-octanoate undergoes the rehydration, meaning it gains a water molecule again, but this time, the water molecule adds to the intermediate in a different position. This arrangement results in isocreate, a 6-carbon molecule with a hydroxyl group OH and the second carbon, forming a secondary alcohol. Step 3, oxidative decarboxylation of isocitrate to alpha-ketoglutarate. In the step 3, isocreate undergoes oxidation and decarboxylation to form a alpha-ketoglutarate, a 5-carbon molecule. Oxidation. First, isocitrate is oxidized by the enzyme isocreate dehydrogenase, which uses NAD+ as an electron acceptor. During this reaction, NAD+ is reduced to NADH, capturing the high-energy electrons for use in the later steps of the cell's energy production pathways. Decarboxylation. Next, the oxidized intermediate loses a carbon dioxide CO2 molecule throughout decarboxylation. This step transforms isocreate into alpha-ketoglutarate, a 5-carbon molecule with a ketone group in place of one of the carboxyl group. This reaction is a crucial regulate steps in the Krebs cycle. It is the both irreversible and the high exogonic release energy, making it the important control point.
Krebs cycle step 4, conversion of alpha-ketoglutarate to succinyl-CoA. In this step, the molecule alpha-ketoglutarate undergoes a reaction with coenzyme A or CoA. Its enzyme is the alpha-ketoglutarate dehydrogenase that facilitates this reaction in step 4. During this process, a carbon atom is removed in the alpha-ketoglutarate and release our carbon dioxide or CO2. Also, the coenzyme NAD+ reduced to form NADH, which store energy for later and used to generate ATP. So its final product is the succinyl-CoA, a high-energy intermediate molecule, and will be later discussed in step 5. To summarize, the enzyme is the alpha-ketoglutarate dehydrogenase. The reactants is the alpha-ketoglutarate, CoA, and the NAD+. The products is the succinyl-CoA, NAD+, and CO2. And lastly, the energy storage is the NAD+. Let's step into the step 5 of conversion of CoA to succinate. So in this step, the succinyl-CoA is converted into succinate, a simpler molecule. The enzyme succinyl-CoA synthetase is responsible for these reactions. So during the process, coenzyme A is released from the succinyl-CoA. Also, a molecule of GDP or guanosine diphosphate is phosphorylated to for GTP or guanosine triphosphate. So GTP can now then transfer its phosphate group to ADP. So now the ADP is forming to ATP, which is the main energy currency of the cell. Also, this is the only step in Krebs cycle where GTP to form ATP is directly generated. To summarize, the enzyme is the succinyl-CoA synthetase. The reactants is the succinyl-CoA, GDP, and PI. The products is the succinate, GTP, and CoA. Lastly, the energy storage is the GDP to ATP.
Step 6, oxidation of succinate to fumarate. In this step, the molecule succinate, which was formed in step 5, is oxidized, meaning it loses electrons. The enzyme that catalyzes this reaction is called succinate dehydrogenase. During deoxidation, succinate donates 2 electrons and 1 proton to the electron carrier FAD, reducing it to FADH2. FADH2 is an important energy carrier molecule that will later contribute to ATP production in the electron transport chain. As a result of this oxidation, succinate is transformed into fumarate, a 4-carbon compound. In step 7, fumarate from step 6 undergoes a hydration reaction, meaning a water molecule is added to it. The enzyme fumarase catalyzes this reaction where water is added to fumarate, converting it into malate, a 4-carbon compound. This step is simply about adding a water molecule to the fumarate, which creates malate. Malate is then ready for the next step in the cycle, where it will be oxidized to produce more energy carriers.
Si Bunga Francesca, Step 8 of Krebs cycle. Krebs cycle has 8 steps. The 8th step is where malate is oxidized to produce oxaloacetate, which is also the starting compound of the citric acid cycle, or the Krebs cycle, and its end compound. During this oxidation, NAD plus is reduced to NADH. The regeneration of oxaloacetate ensures the continuity of the Krebs cycle.
Electron transport chain.
Step 1 of electron transport chain. In mitochondria, energy production starts with NADH, a molecule that carries high energy electrons. When NADH reaches the first complex, NADH dehydrogenase, it releases a hydrogen atom. This hydrogen is made up of 2 parts, a proton, H+, and an electron, which is E negative. As the hydrogen releases its electron, it becomes positively charged. A proton, which is H+. The electron being negatively charged moves through the complex, ready to be passed along the electron transport chain. As electrons move, they are drawn towards areas of positive charge, helping drive the flow of energy. Meanwhile, NADH itself is converted back into NAD+ and sent back to glycolysis and the Krebs cycle to pick up more electrons. Now, the electron moves from NADH dehydrogenase to a mobile carrier called ubiquinone or coenzyme Q. This molecule picks up the electron and carries it to the next complex, cytochrome BC, where the electron will continue its journey through the chain, powering the production of ATP.
Polyester electron complex step 2 and 3. So unlike complex 1, complex 2 doesn't pump protons but deliver electrons directly to ubiquinone. The dual role of complex 2 ensures the integration of metabolic pathways with oxidative phosphorylation, highlighting its importance in maintaining electron flow and efficient energy production. This mechanism reflects mitochondrial adaptability in utilizing diverse fuel sources. The structural details of complex 2 provide insight into electron transfer within the protein complex. So the step 3, the cytochrome BC1 complex and electron transport chain. The electron transport chain, or ATC, generates cellular energy. Complex 3, or cytochrome BC1 complex, is crucial in this chain, transferring electrons from ubiquinol QH2 to cytochrome C and creating the proton gradients for ATP synthesis. So complex 3 mediates electron transfer from QH2 to cytochrome C, pumps protons into the intermembrane space and catalyze the Q cycle, optimizing energy transfer.
Step 4. Electrons transferred to oxygen. Complex 4 receives electrons from cytochrome C and passes them through a series of redox reactions within the complex. These electrons ultimately reduce oxygen, a molecule that is crucial for the chain to function. Oxygen combines with 4 electrons and 4 protons from the mitochondrial matrix to form 2 molecules of water. This reaction is critical as it prevents the buildup of electrons, which would otherwise block the flow through the chain. At the same time, complex 4 pumps protons from the matrix into the intermembrane space. This adds up to the proton gradient across the inner mitochondrial membrane, which is K for ATP synthesis. Step 5. Creating a proton gradient and making ATP. Complex 1, 3, and 4 pump protons from the matrix into intermembrane space as electrons move through the chain. This creates a high concentration of protons in the intermembrane space and a low concentration in the matrix, forming a proton gradient or proton motive force. Protons naturally want to flow back into the matrix to balance the gradient. They do this through ATP synthesis, a protein complex embedded in the inner membrane. As protons flow through ATP synthesis, their movement provides the energy needed to combine ADP and an organic phosphate to form ATP. For every 3 protons that pass through ATP synthesis, 1 ATP molecule is produced. This process, known as chemiosmosis, is the final step in oxidative phosphorylation and is responsible for generating the majority of ATP in cellular respiration.